Welcome to Chapter 2, Section 3, Instruments of Microscopy. In this chapter, the learning objectives are to identify and describe the parts of a Brightfield microscope, calculate total magnification for a compound microscope, and to describe the distinguishing features and typical uses for various types of light microscopes, electron microscopes, and scanning probe microscopes. The early pioneers of microscopy opened a window into the invisible world of microorganisms, but microscopy continued to advance in the centuries that followed. In 1830, Joseph Jackson Lister created an essentially modern light microscope. The 20th century saw the development of microscopes that leveraged non-visible light, such as fluorescence microscopy, which used an ultraviolet light source, and electron microscopy, which uses short wavelength electron beams. These advances led to major improvements in magnification, resolution, and contrast. By comparison, the relatively rudimentary microscopes of van Leeuwenhoek and his contemporaries were far less powerful than even the most basic microscopes in use today. In this section, we will survey the broad range of modern microscopic technology and common applications for each type of microscope. Light microscopy. Many types of microscopes fall under the category of light microscopes, which use light to visualize images. Examples of light microscopes include Brightfield microscopes, Darkfield microscopes, phase contrast microscopes, differential interference contrast microscopes, fluorescence microscopes, confocal scanning laser microscopes, and two-photon microscopes. These various types of light microscopes can be used to complement each other in diagnostics and research. So first up, we have the Brightfield microscopes. The Brightfield microscope, perhaps the most commonly used type of microscope, is a compound microscope with two or more lenses that produce a dark image on a bright background. Some Brightfield microscopes are monocular, having a single eyepiece, though most newer Brightfield microscopes are binocular, having two eyepieces, like the one shown in the figure on the next page. Also, it is not uncommon to see triocular Brightfield microscopes, though generally the third eyepiece is connected to a camera. In either case, each eyepiece contains a lens called an ocular lens. The ocular lenses typically magnify images 10 times, or 10x. At the other end of the body tube are a set of objective lenses on a rotating nosepiece. The magnification of these objective lenses typically range from 4x to 100x, with the magnification for each lens designated on the metal casing of the lens. The ocular and objective lenses work together to create a magnified image. The total magnification is the product of the ocular magnification times the objective magnification, as indicated here. For example, if a 40x objective lens is selected and the ocular lens is 10x, the total magnification would be 40x times 10x equals 400x. Now, the 40x objective is traditionally called the high and dry objective. So the item being viewed is called a specimen. The specimen is placed on a glass slide, which is then clipped into place on the stage, a platform, of the microscope. Once the slide is secured, the specimen on the slide is positioned over the light using the XY mechanical stage knobs, right over here. These knobs move the slide on the surface of the stage but do not raise or lower the stage. Once the specimen is centered over the light, the stage position can be raised or lowered to focus the image. The coarse focusing knob is used for large-scale movement, right here, with the 4x and 10x objective lenses. The fine focusing knob is used for small-scale movements, especially with the 40x and the 100x objective lenses. As you get more experience using these focus knobs, using the coarse focus under 40x for a total of 400x is generally just fine, at least in my experience. When images are magnified, they become dimmer because there is less light per unit area of image. Highly magnified images produced by microscopes, therefore, require intense lighting. In a Brightfield microscope, this light is provided by an illuminator, which will be down here, which is typically a high-intensity bulb below the stage. Light from the illuminator passes up through the condenser lens located below the stage, like directly below it, which focuses all of the light rays on the specimen to maximize illumination. The position of the condenser can be optimized using the attached condenser focus knob. Once the optimal distance is established, the condenser should not be moved to adjust the brightness. If less than maximal light levels are needed, the amount of light striking the specimen can be easily adjusted by opening or closing a diaphragm between the condenser and the specimen. That's this little knob right over here. In some cases, brightness can also be adjusted using a rheostat, a dimmer switch that controls the intensity of the illuminator. This will either be on the side or that might be in the front. It depends on the make and model of the microscope. Generally, as you go up in magnification, you will likely need to increase the light level, so you will adjust the rheostat. A Brightfield microscope creates an image by directing light from the illuminator at the specimen. This light is differentially transmitted, absorbed, reflected, or refracted by different structures. Different colors can behave differently as they interact with chromophores, pigments that absorb and reflect particular wavelengths of light, in parts of the specimen. Often chromophores are artificially added to the specimen using stains, which serve to increase contrast and resolution. In general, structures in the specimen will appear darker, to various extents, than the bright background, creating maximally sharp images at magnifications up to about 1,000 times. Further magnification would create a larger image, but without increased resolution. This allows us to see objects as small as bacteria, which are visible at about 400x or so, but not smaller objects such as viruses. At very high magnifications, resolution may be compromised when light passes through the small amount of air between the specimen and the lens. This is due to the large difference between the refractive indices of air and glass. The air scatters the light rays before they can be focused by the lens. To solve this problem, a drop of oil can be used to fill the space between the specimen and an oil immersion lens, a special lens designed to be used with immersion oils. Since the oil has a refractive index very similar to that of glass, it increases the maximal angle at which light leaving a specimen can strike the lens. This increases the light collected and thus the resolution of the image. A variety of oils can be used for different types of light. Microconnections, microscopic maintenance best practices. Even a very powerful microscope cannot deliver high-resolution images if it is not properly cleaned and maintained. Since lenses are carefully designed and manufactured to refract light with a high degree of precision, even a slightly dirty or scratched lens will refract light in unintended ways, degrading the image of the specimen. In addition, microscopes are rather delicate instruments, and great care must be taken to avoid damaging parts and surfaces. Among other things, proper care of a microscope includes the following: cleaning the lenses with lens paper, not allowing lenses to contact the slide by rapidly changing the focus, protecting the bulb, if there is one, from breakage. One aspect of protecting the bulb is to start at the lowest illumination level possible and then increasing the light level as you need, and then returning it back to 1 or 0, depending on the model, when you are done viewing. Not pushing an objective into a slide. The 40x and the 100x will most likely be spring-loaded. This protects you a little bit, but it's best just not to have the lens contact the slide in the first place. Not using the coarse focus knob when using the 40x or greater objective lenses. Only using immersion oil with a specialized oil objective, usually the 100x objective. Cleaning oil from immersion lenses after using the microscope. This is very important because it is not uncommon for oil to get dragged back to the 40x objective. Cleaning any oil accidentally transferred from other lenses, covering the microscope, or placing it in a cabinet when not in use. Okay, next, Darkfield Microscopy. A darkfield microscope is a brightfield microscope that has a small but significant modification to the condenser. A small opaque disc, about 1 centimeter in diameter, is placed between the illuminator and the condenser lens. This opaque light stop, as the disc is called, blocks most of the light from the illuminator as it passes through the condenser on its way to the objective lens, producing a hollow cone of light that is focused on the specimen. Only light that reaches the objective is light that has been refracted or reflected by structures in the specimen. The resulting image typically shows bright objects on a dark background. Darkfield microscopy can often create high contrast, high resolution images of specimens without the use of stains, which is particularly useful for viewing live specimens that might be killed or otherwise compromised by the stains. For example, thin spirochetes like Treponema pallidum, the causative agent of syphilis, can be best viewed using a darkfield microscope. So, clinical focus, part 2. Wound infections like Cindy's can be caused by many different types of bacteria, some of which can spread rapidly with serious complications. Identifying the specific cause is very important to select a medication that can kill or stop the growth of bacteria. After calling a local doctor about Cindy's case, the camp nurse sends the sample from the wound to the closest medical laboratory. Unfortunately, since the camp is in a remote area, the nearest lab is small and poorly equipped. A more modern lab would likely use other methods to culture, grow, and identify the bacteria, but in this case, the technician decides to make a wet mount from the specimen and view it under a Brightfield microscope. In a wet mount, a small drop of water is added to the slide, and a cover slip is placed over the specimen to keep it in place before it is positioned under the objective lens. Under the Brightfield microscope, the technician can barely see the bacterial cells because they are nearly transparent against the bright background. To increase contrast, the technician inserts an opaque light stop above the illuminator. The resulting darkfield image clearly shows that the bacterial cells are spherical and grouped in clusters like grapes. Why is it important to identify the shape and growth patterns of cells in a specimen? What other types of microscopy could be used to effectively view this specimen? Phase contrast microscopes. Phase contrast microscopes use refraction and interference caused by structures in a specimen to create high contrast, high resolution images without staining. It is the oldest and simplest type of microscope that creates an image by altering the wavelengths of light rays passing through the specimen. To create altered wavelength paths, an annular stop is used in the condenser. The annular stop produces a hollow cone of light that is focused on the specimen before reaching the objective lens. The objective contains a phase plate containing a phase ring. As a result, light traveling directly from the illuminator passes through the phase ring, while light refracted or reflected by the specimen passes through the plate. This causes waves traveling through the ring to be about 1/2 of a wavelength out of phase with those passing through the plate. Because waves have peaks and troughs, they can add together, if in phase together, or cancel each other out, if out of phase. When the wavelengths are out of phase, wave troughs will cancel out wave peaks, which is called destructive interference. Structures that refract light then appear dark against a bright background of only unrefracted light. More generally, structures that differ in features such as refractive index will differ in levels of darkness. So this is the light path, and then this is the kind of image that is produced. Because it increases contrast without requiring stains, phase contrast microscopy is often used to observe live specimens. Certain structures, such as organelles in eukaryotic cells and endospores in prokaryotic cells, are especially well visualized with phase contrast microscopy. Differential interference contrast microscopes. Differential interference contrast DIC microscopes, also known as Nomarski optics, are similar to phase contrast microscopes in that they use interference patterns to enhance contrast between different features of a specimen. In a DIC microscope, two beams of light are created in which the direction of wave movement, polarization, differs. Once the beam passes through either the specimen or specimen-free space, they are recombined, and effects of these specimens cause differences in the interference patterns generated by the combining of the beams. This results in high contrast images of living organisms with a three-dimensional appearance. These microscopes are especially useful in distinguishing structures within live, unstained specimens. This is an indication of the light path from Wikipedia. A fluorescence microscope uses fluorescent chromophores called fluorochromes, which are capable of absorbing energy from a light source and then emitting this energy as visible light. Fluorochromes include naturally fluorescent substances, such as chlorophylls, as well as fluorescent stains that are added to the specimen to create contrast. Dyes such as Texas Red and FITC are examples of fluorochromes. Other examples include the nucleic acid dyes 4,6-diaminido, 2-phenylindole, DAPI, and acridine orange. The microscope transmits an excitation light, generally a form of electromagnetic radiation with a short wavelength, such as ultraviolet or blue light, towards the specimen. The chromophores absorb the excitation light and emit visible light with longer wavelengths, or the fluorophores. The excitation light is then filtered out, in part because ultraviolet light is harmful to the eyes, so that only visible light passes through the ocular lens. This produces an image of the specimen in bright colors against a dark background. Fluorescence microscopes are especially useful in clinical microbiology. They can be used to identify pathogens, to find particular species within an environment, or to find the locations of particular molecules and structures within a cell. Approaches have also been developed to distinguish living from dead cells using fluorescence microscopy based upon whether they take up a particular fluorochrome. Sometimes multiple fluorochromes are used on the same specimen to show different structures or features. One of the most important applications of fluorescence microscopy is a technique called immunofluorescence, which is used to identify certain disease-causing microbes by observing whether antibodies bind to them. Antibodies are protein molecules produced by the immune system that attach to specific pathogens to kill or inhibit them. There are two approaches to this technique: direct immunofluorescence assay, DFA, and indirect immunofluorescence assay, IFA. In DFA, specific antibodies, for example, those that target the rabies virus, are stained with a fluorochrome. If the specimen contains the targeted pathogen, one can observe the antibodies binding to the pathogen under the fluorescent microscope. This is called a primary antibody stain because the stained antibodies attach directly to the pathogen. See, any antibody that did not bind is washed away. In IFA, secondary antibodies are stained with a fluorochrome rather than the primary antibodies. Secondary antibodies do not attach directly to the pathogen, but they do bind to primary antibodies, so they are an antibody-antibody. When the unstained primary antibodies bind to the pathogen, the fluorescent secondary antibodies can be observed binding to the primary antibodies. Thus, the secondary antibodies are attached indirectly to the pathogen. Since multiple secondary antibodies can often attach to a primary antibody, IFA increases the number of fluorescent antibodies attached to the specimen, making it easier to visualize features in the specimen. Confocal microscopes. Whereas other forms of light microscopy create an image that is maximally focused at a single distance from the observer, the depth or the z-plane, a confocal microscope uses a laser to scan multiple z-planes successively. This produces numerous two-dimensional high-resolution images at various depths, which can be constructed into a three-dimensional image by a computer. As with fluorescence microscopes, fluorescent stains are generally used to increase contrast and resolution. Image clarity is further enhanced by a narrow aperture that eliminates any light that is not from the Z-plane. Confocal microscopes are thus very useful for examining thick specimens such as biofilms, which can be examined alive and unfixed. Two-photon microscopes. While the original fluorescent and confocal microscopes allowed better visualization of unique features in specimens, there were still problems that prevented optimum visualization. The effective sensitivity of fluorescence microscopy when viewing thick specimens was generally limited by out-of-focus flare, which resulted in poor resolution. This limitation was greatly reduced in the confocal microscope through the use of a confocal pinhole to reject out-of-focus background fluorescence with thin, less than 1 micrometer, unblurred optical sections. However, even the confocal microscope lacked the resolution needed for viewing thick tissue samples. These problems were resolved with the development of the two-photon microscope, which uses a scanning technique, fluorochromes, and long wavelength light, such as infrared, to visualize specimens. The low energy associated with the long wavelength light means that two photons must strike a location at the same time to excite the fluorochrome. The low energy of the excitation light is less damaging to cells, and the long wavelength of the excitation light more easily penetrates deep into thick specimens. This makes the two-photon microscope useful for examining living cells within intact tissues, brain slices, embryos, whole organs, and even entire animals. Currently, the use of two-photon microscopes is limited to advanced clinical and research laboratories because of the high costs of the instruments. A single two-photon microscope typically costs between $300,000 and $500,000, and the lasers used to excite the dyes used on specimens are also very expensive. However, as technology improves, two-photon microscopes may become more readily available in clinical settings. Electron microscopy. The maximum theoretical resolution of images created by light microscopes is ultimately limited by the wavelength of visible light. Most light microscopes can only magnify 1,000 times, and a few can magnify up to 1,500 times, but this does not begin to approach the magnifying power of an electron microscope, which uses short wavelength electron beams rather than light to increase magnification and resolution. Electrons, like electromagnetic radiation, can behave as waves, but with wavelengths of 0.005 nanometers, they can produce much better resolution than visible light. An electron microscope can produce a sharp image that is magnified up to 100,000 times. Thus, EMs can resolve subcellular structures as well as some molecular structures, single strands of DNA. However, electron microscopy cannot be used on living material because of the methods needed to prepare the specimens. There are two basic types of electron microscopes: the transmission electron microscope, TEM, and the scanning electron microscope, SEM. The TEM is somewhat analogous to the Brightfield Light Microscope in terms of the way it functions. However, it uses an electron beam from above the specimen that is focused using magnetic lenses rather than a glass lens and projected through the specimen onto a detector. Electrons pass through the specimen, then the detector captures the image. However, it uses an electron beam from above the specimen that is focused using a magnetic lens rather than a glass lens and projected through the specimen onto a detector. Electrons pass through the specimen and the detector captures the image. For electrons to pass through the specimen in a TEM, the specimen must be extremely thin, 20 to 100 nanometers thick. The image is produced because the varying opacities in various parts of the specimen. This opacity can be enhanced by staining the specimen with materials such as heavy metals, which are electron dense. TEM requires that the beam and specimen be in a vacuum and that the specimen be very thin and dehydrated. The specific steps needed to prepare a specimen for observation under an EM are discussed in detail in the next section. SEMs form images of surfaces of specimens, usually from electrons that are knocked off a specimen by a beam of electrons. This can create highly detailed images with a three-dimensional appearance that are displayed on a monitor. Typically, specimens are dried and prepared with fixatives that reduce artifacts, such as shriveling, that can be produced by drying, before being sputter-coated with a thin layer of metal such as gold. Whereas transmission electron microscopy requires very thin sections and allows one to see internal structures, such as organelles and the interior of membranes, scanning electron microscopy can be used to view the surfaces of larger objects, such as a pollen grain, as well as the surfaces of very small samples. Some electron microscopes can magnify an image up to 2 million times. Microconnections, using microscopy to study biofilms. A biofilm is a complex community of one or more microorganism species, typically forming as a slimy coating attached to a surface because of the production of an extrapolymeric substance, EPS, that attaches to a surface or at the interface between surfaces, like between air and water. In nature, biofilms are abundant and frequently occupy complex niches within ecosystems. In medicine, biofilms can coat medical devices and exist within the body. Because they possess unique characteristics such as increased resistance against the immune system and to antimicrobial drugs, biofilms are of particular interest to microbiologists and clinicians alike. Because biofilms are thick, they cannot be observed very well using light microscopy. Slicing a biofilm to create a thinner specimen might kill or disturb the microbial community. Confocal microscopy provides clearer images of biofilms because it can focus on one Z-plane at a time and produce a three-dimensional image of a thick specimen. Fluorescent dyes can be helpful in identifying cells within the matrix. Additionally, techniques such as immunofluorescence and fluorescence in situ hybridization, or FISH, in which fluorescent probes are used to bind to DNA, can be used. Electron microscopy can be used to observe biofilms, but only after dehydrating the specimen, which produces undesirable artifacts and distorts the specimen. In addition to these approaches, it is possible to follow water currents through the shapes, such as cones and mushrooms, of biofilms, using video of the movement of fluorescently coated beads. Scanning probe microscopy. A scanning probe microscope does not use light or electrons, but rather very sharp probes that are passed over the surface of the specimen and interact with it directly. This produces information that can be assembled into images with magnifications up to 100 million times. Such large magnifications can be used to observe individual atoms on surfaces. To date, these techniques have been used primarily for research rather than for diagnostics. There are two types of scanning probe microscopes: the scanning tunneling microscope, STM, and the atomic force microscope, AFM. An STM uses a probe that is passed just above the specimen at a constant voltage bias, creates the potential for an electric current between the probe and the specimen. This current occurs via quantum tunneling of electrons between the probe and the specimen, and the intensity of the current is dependent upon the distance between the probe and the specimen. The probe is moved horizontally above the surface, and the intensity of the current is measured. Scanning tunneling microscopy can effectively map the structures of surfaces at a resolution at which individual atoms can be detected. Similar to an STM, AFMs have a thin probe that is passed just above the specimen. However, rather than measuring variations in the current at a constant height above the specimen, an AFM establishes a constant current and measures variation in the height of the probe tip as it passes over the specimen. As the probe tip is passed over the specimen, forces between the atoms, van der Waals forces, capillary forces, chemical bonding, electrostatic forces, and others, cause it to move up and down. Deflection of the probe tip is determined and measured using Hooke's law of elasticity, and this information is used to construct images of the surface of the specimen with resolution at the atomic level. And these figures summarize the features of the various microscopes discussed herein. So the Brightfield microscope is commonly used in a wide variety of laboratory applications, as the standard microscope produces an image on a bright background. The Darkfield microscope increases contrast without staining by producing a bright image on a darker background, especially useful for viewing live specimens. Phase contrast uses refraction and interference caused by structures in the specimen to create high contrast, high resolution images without staining, making it useful for viewing live specimens and structures such as endospores and organelles. Differential interference contrast uses interference patterns to enhance contrast between different features of a specimen to produce high contrast images of living organisms with a three-dimensional appearance, making it especially useful in distinguishing structures within live, unstained specimens. Images viewed reveal detailed structures within cells. Fluorescence uses fluorescent stains to produce an image, can be used to identify pathogens, to find particular species, to distinguish living from dead cells, or to find locations of particular molecules within a cell, also used for immunofluorescence. Confocal uses a laser to scan multiple Z-planes successfully, producing numerous two-dimensional high-resolution images at various depths that can be constructed in a three-dimensional image by a computer, making this useful for examining thick specimens such as biofilms. 2-photon uses a scanning technique, fluorochromes, long wavelength light, such as infrared, to penetrate deep into thick specimens such as biofilms. So for electron microscopes, we're using an electron beam focused with magnets to produce an image. The transmission electron microscope uses electron beams that pass through a specimen to visualize small images, useful to observe small, thin specimens such as tissue selections and subcellular structures. The scanning electron microscope uses electron beams to visualize surfaces, useful to observe the three-dimensional surface details of specimens. Scanning probe microscopes, we're getting it up into magnification of 100 million times or more. The scanning tunneling microscope uses a probe passed horizontally at a constant distance just above the specimen while the intensity of the current is measured, can map the structures of surfaces at the atomic level, works best on conducting materials, but can also be used to examine organic materials such as DNA if fixed on a surface. Atomic force microscopes can be used in several ways, including using a laser focused on a cantilever to measure the bending of the tip or a probe passed above the specimen while the height needed to maintain a constant current is measured. Useful to observe specimens at the atomic level and can be more easily used with non-conducting samples. Okay, that brings us to the end of section 3. Join me next time for section 4, staining microscopic specimens. Until then.